Welcome to the Ethel Browne Harvey Postdoctoral Seminar Series. My name is Casey Griffin, and I'm a postdoc at New York University, and will be moderating today with Martha Echevarria, a postdoc at University of Wisconsin-Madison. We are excited to highlight the work of our outstanding postdoctoral members. Today, Yuchuan Miao from Harvard Medical School and Brigham and Women's Hospital, and Kari Price from Yale University School of Medicine will share their research. Each speaker will give a 20-minute talk followed by 10 minutes of Q&A. Please enter your questions in the Zoom Q&A box. We will begin with Yuchuan Miao. Yuchuan got his Bachelor of Science in Chemistry from Nankai University in Tianjin, China in 2011. He then completed his PhD in biochemistry and cell biology from Johns Hopkins University School of Medicine in 2019. Yuchuan started his postdoc at Harvard Medical School and Brigham and Women's Hospital in 2019 in the lab of Olivier Porquier. He has received numerous honors and awards, including the Top 3 Trainee Award from Harvard Stem Cell Institute, the Rising Stars in Engineering and Health from Columbia and Johns Hopkins University, and a K99 Pathway to Independence Award from the NIH. Yuchuan will be talking today about reconstruction and deconstruction of human somatogenesis in vitro. Thank you, Yuchuan.
Yeah, thank you so much for the kind introduction. And thank you for the opportunity for me to share my work here. Let me get started. Okay, my name is Yuchuan, and I'm interested in how multicellular systems make patterns and shapes during human development. As a cell biologist by training, I'm especially interested in cell biological level mechanism of pattern formation. In my postdoc, I was focusing on the fascinating process of somatogenesis, or somites formation in the early embryo. So somites are the repeated epithelial blocks flanking our neural tube. They give rise to our axial skeleton and skeletal muscle and so on. They are instrumental to establish the segmental features of our body plan. In all vertebrates, somatogenesis share several conserved features. Very interestingly, somites are not formed all at once. They are formed sequentially from head to tail. Each somite will bud off from an unsegmented region called the PSM, one by one, in coordination with the elongation of the body axis. And the rhythmicity of somite formation is controlled by a conserved oscillator of gene regulatory network called a segmentation clock. In each cycle, the clock activities denoted as blue in the movie cartoon here, will start from the posterior end and propagating anteriorly as a wave. And when they reach the anterior region, they together with signal ingredients specify a newly forming somites. Now, extensive genetic studies have identified that Has7 is a core gene of the segmentation clock in mouse and human, while the MESP2 gene in the expression of MESP2 in the anterior region labels the onset of somite formation. In addition, somites are also intricately patterned. Each somite is divided into an anterior and posterior half, characterized with specific gene transcriptome as well as developmental trajectories. One of the genes UNCs labels the posterior half of each somite. If we look at the UNC's RNA expression in mouse embryo, you can see that they form these beautiful periodic stripes along our body axis. Now, this somite AP polarity patterning, also called Rose-Trocado patterning, is extremely important to the development of our proper development, our body plan. They instruct the formation of our vertebrae of our spine, and also because only the anterior half of the somite permits the migration of axons, they confers the segmental organization to our peripheral nervous system as well. So extensive study using animal embryos have allowed us to gain lots of knowledge about somatogenesis, but there are still few challenges in further understanding somatogenesis. So although the segmentation clock control the racemative somites formation has begun to be well characterized, but the subsequent somite patterning and morphogenesis and their connection with the clock are less understood. And among all of our knowledge, we have a much less understanding on the cellular level mechanisms of controlling this process. Most of the classical studies focusing on tissue level gene expression. After all, it's the cells that build these beautiful patterns and shapes. And furthermore, we have extremely little about the knowledge of this important process in our own species. In humans, somites occurs very early during development, between 3 to 4 weeks after fertilization. It's a time window when most women haven't realized their pregnancy yet. Thus, we have extremely limited access to human embryos at this stage. So to address these challenges, today I'm showing you some of my work using stem cell-based approaches to address these challenges. I will use human pluripotent stem cells to build 3D organoid models that recapitulate the patterning and morphogenesis process of somites formation. These will allow us to directly study human development, and also they allow direct visualization, much easier visualization and sophisticated perturbation of cellular activities in these processes. And then I will show you some of the insights we have gained by using these in vitro models to study somite patterning. Okay, so long story short, I have built 2 models to recapitulate somite formation. The first model, we call them somitoid. This is model recapitulate somite formation in time. In the embryo, a group of cells will periodically undergo differentiation and morphogenesis. And in the model, somitoid model, all the cells undergo this differentiation trajectory synchronously. As shown by this video here with the reported cell lines, you can see that the cells go into this oscillation phase of Has7, and then they altogether go into this segmental determinant phase labeled by MSP2, and then starting to go to forming these rosette or somite-like structures, and eventually forming a field of somite-like structures. So each of these rosettes has the characteristics of somites, including the epithelial blocks, as well as surrounded by a laminin sheet. So somitoid is a model that only have one axis time that all the cells synchronously differentiate. So it's a simplified model of somatogenesis. Now, the second model I have built is called segmentoid. So this model recaptures both time and space during somite formation. So if I play this video, you will see that oscillatory expression of the Has7 will occur at the posterior end as the structure elongates, while the segmental gene MSP2 follows suit. And eventually, they give rise to these rod-shaped-like structures with somite-like rosettes, as well as recapitulate the anterior-posterior patterning of each row of somites, as labeled by the stripes expression of Ankh here. Yeah, so together with several other models built by other labs, we provide a platform to allow us to study human somatogenesis. Between the 2 models I have built, they each have their own advantages. For somitoid, it's a situation where all the cells are precisely synchronized. So we have unlimited amount of cells at each time point for in-depth characterization. Their configuration is also amenable for higher resolution imaging to look at the cellular activities. On the other hand, segmentoid is embedded in matrigel, but they display all these important hallmarks of somatogenesis can be used as a proxy to test these insights we have gained from using somitoid. So as I mentioned earlier, I'm really interested in the patterning and morphogenesis process. So the question I have starting to use my system to address is my anterior posterior polarity patterning. In this process, much of our understanding about this process comes from the extensive work by Yumiko Saga. And it is observed that MSP2, the segmental gene, is first inducted uniformly across the whole length of the newly formed segments. And then MSP2 expression is restricted to the anterior portion. And because MSP2 is a repressor of notch signaling, so in the anterior half of the somite, we're experiencing lower notch signaling. While on the posterior half, in addition with further applications, it experiences higher notch signaling. Thus, this differential notch signaling really determines anterior or posterior fate maturation. However, what is not known is how is the initial symmetry breaking process established? How is MSP2 transition from this broadband expression to the only half-band expression? It is perceived that this process is a process of a tissue-level gene remodeling process, that somehow the posterior part, MSP2 expression is suppressed. However, there are no concrete models for this perception. Then I decided to use my models to look at in detail how this process is established. I first turned to my somatoid model. This is really interesting because somatoid is a simplified model, and it challenges this whole system in a completely new context that does not exist naturally. If you think about this model doesn't really have anterior posterior information, how would the patterning be like in this model? And much to our surprise, we found that not only can anterior posterior fated cells come up, but also they form into patterns. That in this model, is formed mostly entire somewhat-like structures with entirely anterior or posterior fates labeled by UNCS and MSP2 here. This is extremely interesting. So I decided to use live imaging to look at how this process is occurred. So this movie is going to showing you the time window labeled as the blue box here, as the green box here. So it's the time window when MSP2 is transiently expressed. On the left is the MSP2 reporter. You will see is that the signaling comes up, but then at the end of time, like 72 hours, when MSP2 intensity is not increasing anymore, they ended up in this salt and pepper fashion. That not all cells express that. The cells express like have the difference in MSP2 expression as big as up to 10-fold. This directly contracts our traditional notion of a uniform MSP2 expression in a forming segment. And if you follow this movie a little bit longer, right now is labeled by this blue box, you will see is that there are extensive cell movement, and then MSP2 high cells starting to aggregate, while MSP2 low cells starting to come together, eventually forming these different clusters of MSP2 high and MSP2 low regions. Now here's a movie with a further labeling to see this process. The orange line outlines the future MSP2 low region, while the color cells labels MSP2 high cells. You can see that the cells starting randomly, but then they gradually have moved out of this region. And in the end, they starting to form MSP2 low regions after all these MSP2 high cells have moved out. Thus, it says that the cells, it seems that these cell fates are first determined before any patterns has formed. So to confirm this, we decided to using dissociation and reaggregation assays. So we take cells at 72 hours. Remember, this is a time when MSP2 has finished its expression. It's ended up in a salt and pepper fashion. There are no pattern formed yet. And then we dissociate the somatoids completely into single cells. And then using FACS to separate the top 10 MSP2 high cells and bottom 10 MSP2 low cells. and re-aggregatism, just centrifugation. And we continue to culture them. And by 120 hours, we want to see their expression profile of UNCs, the mature gene that labels the posterior fates. Now, interestingly, you can see is that the MASP2 high cells isolated at the salt pepper no pattern stage, they will continue to express lower level of UNCs, while MASP2 low cells at this earlier stage will express much more UNCs expression. So this suggesting that indeed the cell identities are already determined at the salt and pepper stage before any patterns has formed. So all of these, together with some other quantifications, led to a sequence of events that we think what's happened in somatoid is that the MASP2 is first inducted in salt and pepper fashion. And importantly, at this stage, the anterior posterior fate in each cells is more or less already determined. And then through cell sorting, they quickly form differential clusters. And as time goes on, each rosettes emerge from each clusters. As you can see that the cluster can be much bigger than the typical somite length. So that's why in the end, we end up with somites of entirely anterior or posterior fates. So what does this mean for what happens in embryos? Could it be that the conventionally thought broadband, narrowband expression actually be a result of cell sorting? So we further test this using the other model, segmentoid, to use as embryo proxy. So in this video with the red box label a forming segment, in the middle, you can see that in this forming segment, MASP2 is already inducted and in a salt and pepper fashion. And then as time goes on, you can see there extensive cell movement, and eventually the MASP2 high cells starting to cluster and low cells starting to cluster, eventually forming differential compartments of anterior or posterior fates. And then here's a video, further zoomed in video to look at this process. On the left is unlabeled, on the right is labeled. In this forming segment region, we can start looking at these blue cells, or these cells corresponding to here. You can see that it inducted in the posterior half, but then as this somites develops, is gradually starting to move anteriorly towards the anterior half. So indeed, this is suggesting that the conventionally thought gene remodeling, tissue level gene modeling process might actually is a result of a cell sorting or cell rearrangement. And furthermore, when we using when we inducibly overexpress TM1, which is RAC1 gap, which perturbs the cell skeletal properties, we can completely block cell sorting. And then you can see that there's still oscillation of the clock, but then eventually you can there will be no organized stripes expression formed in the end. So all of these together leads us to a general framework integrating cell sorting, cell act dynamics with the segmentation clock and fate specification. I don't have time to elaborate this model, but if you're interested, you can check out our published work for this general framework. Now, in the end, I just want to say that I just hope I convince you that the power of in vitro systems in uncovering novel mechanism of development. These models are not perfect, but we can use them to dissect molecular, especially cellular mechanism in detail. And the goal of the field is not to build embryos. The goal is to understand biology. And I'm particularly interested in using the system I have built to understand the cellular mechanism of the patterning and morphogenesis during somatogenesis. And on the other hand, I'm also interested in continue building 3D models of modeling different aspects of human development. So with this, I like to thank my mentor Olivier and other collaborators and the whole Pookie lab and our collaborator Stefano D'Italia, whose work I did not have the time to show today. And also thank my funding source And thank you so much for your attention, and I'll be up for questions.
Thank you, Yuchuan. While we wait for questions to open and come in, I will start off. What do you think is the mechanism behind this cell sorting?
Yeah, so that is a continuing work. But we did do a RNA-seq to compare the transcriptome between MES2 high and MES2 low cells. And we found quite a lot of interesting differences involving several adhesion proteins, such as PCDH, protocadherin 8, and also several afferent proteins, and a lot of cell skeleton regulatory proteins. So this is continually exciting work going on, but I think it's just that they are sorted apart through the differential adhesion as well as differential cell skeleton properties.
Great, thank you. So we have a couple questions coming in. First off, Chaitra Prabhakara says, an excellent talk, Yuchuan. I have a question. Are patterns formed in somatoids with just MESP2 high as well as MESP2 low cells?
Yes, so at this stage, so the somite anterior posterior fate is firstly specified MESP2 expression levels. At this stage, MESP2 high cell will specify future anterior face, MESP2 low cell will specify future low cells. And then as the continuing maturation, further mature genes such as UNCs will come up to label more posterior fate. Yeah, so at this stage, it's solely separated through MESP2 differential expression.
Great. Hao Young Wei says, very cool talk. I wonder if you sort the MESP2 high and low cells and reseed them in a certain spatial pattern that is different from the normal somite organization, will the predetermined fate be preserved and cell sorting happening?
Thank you for the question. Let me try to understand reseed in certain spatial pattern that different from the normal somite. Yeah, so this is a very interesting point. I haven't explored this too much yet, but right now what I did is simply sort them apart and re-aggregate them together. I assume that by playing with the density or geometry, we can potentially introduce more perturbations. They actually might be interesting to show what happens for example, to test the requirements for cell sorting in terms of density, in terms of geometry, and so on. Yeah, so that'll be a very interesting point going forward.
Great. Natalia Batasso Arias says, very interesting. Congratulations for your work. Do you know if the segmentoids show signs of left-right patterning?
I don't know right now. We haven't looked at it yet. But I want to point out that in our model so far, it's only consists of the peroxisome mesoderm. So some of the patterning, such as I presume you're asking about left to right or other axes, they might need the signaling inputs from other germ layers. In so far, we don't have it. But that does not mean this axis establishment could not be a self-organized process. It's certainly something interesting to further look at. Thank you.
Wei Shu says, enjoyed your talk. Are there any human diseases caused by defects in oscillation of the segmentation clock? So your human use your models to examine the underlying mechanisms.
Yes, that's actually an exciting direction we're going to, because a lot of the segmental defects of human embryo are caused by players involved in the segmentation clock and segmental genes. So for example, HAS7 and DLL1, these are all genes involved in human segmentation clock. Their mutation always cause severe segmentation defects our spine. So these models can be a very interesting way to further look at the molecular and cellular details of the etiology here.
Martin Arostegue says, great talk and work. Have you noticed any cells begin to delaminate or migrate out of the somatoids or segmentoids?
Yeah, so it depends on time. If you culture these models like longer time, all the cell was starting to migrate out. And also I want to point out I think it's especially interesting for somatoids because at the edge, where the cell layer are usually much thinner than in the center. So at the edge, they don't form rosettes. So there seems to be interesting to look at the edge of how the cells behavior, cell behavior at the edge to tell us more about the rosette formation. But at this stage, they usually tend to stay in this rosette structure as well as there's a certain cell density.
Loic Fort says, great talk. The MESP2 sorting by expression level reminds me of a similar event during intestine specification and folding, driven by PDGFR high versus low. In this system, they show it is dependent on actomyosin, which might be reminiscent with your Team 1 data. Have you tried to abrogate cell contractility in this system?
Yes, we have tried to using ROCK inhibitor to add it into the cell culture here. With the in the presence of ROCK inhibitor, they cannot form rosette-like structures. However, in the presence of ROCK inhibitor, they can still show some rudimentary cell clustering process. So I think it's so my perception is that the cell contractility here, perturbing it with ROCK inhibitor might change the robustness of pattern formation, but it doesn't seem to be essential for the sorting process.
Great. And Hao Young Wei asks again, may I ask if the left side and right side of somite are physically separated? If so, how does the somite formation synchronize between left and right?
The left side and right side of somite physically separated. I think what you mean is each row of the somites in the segmentoid. Yes, they are. If we do a staining, you can see they're indeed separated by ECM. And also we've found that when we seed this, when we starting with a bigger cell aggregate, we will form a segment structure that's much wider, usually consists of 3 or 4 somites. So it seems that in all these cases, the one row perpendicular to the AP axis are always synchronized. We don't know exactly how, but I think it have to do with the segmentation clock and how it's the propagation direction. And yeah, it will be like investigated further in the future.
Great. That's all the questions we have in the box. So with that, I think we will move on.
Thank you so much.
Hi, everyone. It's my pleasure to introduce our next speaker, Kari Price. Kari completed her training at the University of California, Davis in the laboratory of Dr. Leslie Rose, and then she transitioned to a postdoc at the Yale School of Medicine in the laboratory of Dr. Lynn Cooley. Her postdoctoral research focuses on elucidating the mechanism of intracellular bridge formation in animal germline cells. Her research has been supported by an NIH F32 and the Yale Venture Fund and CERTNA Foundation Postdoctoral Fellowship. Today, her presentation is entitled Bridging the Gap, Connecting Cells Through Intracellular Bridges. So, Kari, you can go ahead.
Okay. Play. All right. Thank you, Martha, for the introduction, and thank you, SDB, for the opportunity to present my work here today. And my research focuses on trying to understand how the canonical program of cytokinesis is modified in certain cell types so that following incomplete cytokinesis, these cells remain connected by intercellular bridges. And in most animal cells, cytokinesis is complete and produces 2 physically separate daughter cells with every cell division. However, a number of cell types undergo or utilize a highly regulated program of incomplete cytokinesis that gives rise to cells connected by intercellular bridges. And the formation of these intercellular bridges allows these cells to develop, in effect, as syncytia. And intercellular bridges have been observed in cells with high developmental potential, including embryonic cells and multipotent stem cells, as well as in terminally differentiated cells and clonal multicellular animals. But intercellular bridges are most prominent in the context of the animal germline, where intercellular bridges and their formation is absolutely critical to the formation of male and female germ cells. And in the germline, these intercellular bridges are called ring canals, aptly named because they take on this ring-shaped appearance. And this ring-shaped cytoskeletal structure maintains the cytoplasmic opening connecting these sibling cells. And on the ultrastructural level, ring canals can be easily identified by the presence of this electron-dense plasma membrane that marks the cytoplasm, or the membrane between connecting these 2 cells. And I'm showing you here an example of 2 spermatocytes that are connected by a single cytoplasmic bridge. And marking here with the red arrows, I'm denoting the boundary of this electron density. And we've now generated FibSim data sets, and we can reconstruct these data sets to actually visualize the ring canals in 3D. And I'm showing you here just a reconstruction of this data set where you can more easily see the ring canal structure. Ring canal formation is an incredibly well-conserved feature of gametogenesis, and kinesin 6 is not only a component of the central spindling complex, an important cytokinesis regulator, but it's also a universal ring canal component. And I should mention that ring canals are absolutely critical to the formation of germ cells, as they allow the transfer of mRNAs, proteins, and even organelles between sibling cells. And when we look with this kinesin 6, we could actually see that across the animal kingdom, kinesin 6 localizes robustly to these ring canals. And I'm showing you here just a panel of male germlines from the respective species, where I have taken these germlines and immunolabeled with an antibody that recognizes the kinesin 6 ortholog in each of these species. And so it's this localization of this critical cytokinesis component to ring canals that has hinted at this really intimate relationship between programs of cytokinesis and ring canal formation. And for most of my talk, I am going to be referring to the Drosophila ortholog of kinesin 6, which is a protein that we call Pavarotti or POV. Typically, at the end of the cell cycle, cytokinesis terminates with a mid-body mediated abscission, which physically separates these nascent daughter cells. And during cytokinesis, an actomyosin contractile ring assembles at the equatorial cortex between the segregating sister chromatids, and does so in response to cues from the central spindle and central spindolin, which is a heterotetrameric complex of MGC RAC gap and kinesin 6, its obligate binding partner. Following constriction of the actomyosin contractile ring, a midbody forms, which marks this transient intercellular bridge, and its architecture can be subdivided into 2 groups. The midbody core, shown here in purple, which is an accumulation of central spindolin protein, and the midbody ring, which is comprised of former components of the contractile ring, including anilin, citron kinase, and the septins. And the midbody plays an important role in mediating cytokinesis, as it serves as a platform for the recruitment of the abscission machinery, which is necessary to sever this transient cytoplasmic bridge, physically separating and permanently separating these newly synthesized daughter cells. And following division, a midbody remnant forms. And this midbody remnant, depending on the cell context, can either be absorbed by one of the daughter cells or extruded into the medium. However, in contrast, during incomplete cytokinesis, this process is marked by a characteristic absence of abscission, and there's been much work that has been done to try and understand the mechanism inhibiting abscission. But it remains unclear exactly how cytokinesis is altered in the dividing germ cell. In my research, and I am particularly interested in understanding how the process of abscission inhibition is coordinated with the cytoskeletal changes that underlie ring canal formation, and specifically how components that localize to the central spindle and to the contractile ring come to be localized into these membrane-bound, stable ring canal structures. And I use the Drosophila melanogaster testis as my model system, where spermatogenesis occurs in a spatiotemporal pattern. And at the apical tip of the testis, the germline stem cells and the somatic stem cells surround a cluster of somatic cells called the hub. The germline stem cell will divide asymmetrically to produce a gonioblast and another germline stem cell. The gonioblast then goes on to divide 4 times mitotically and 2 times meiotically to generate 64 haploid sperm. And with each mitotic and meiotic division, cytokinesis is incomplete, resulting in the formation of a single ring canal. And in contrast, the somatic stem cell divides completely to generate a somatic stem cell and a somatic cyst cell that will envelop the gonioblast and will surround the developing germ cell throughout the entire process of spermatogenesis. Another feature that's important to my work is this structure called the fusum, which is shown here in this translucent gray. And it serves as another form of connectivity between cells, sibling cells within a cyst. And the fusum is this branched-like structure that extends throughout all of the ring canal lumens that connect all of the cells in a germline cyst. And when I first joined the Cooley Lab, I really wanted to characterize ring canal formation in real time with high spatiotemporal live imaging. And the first thing that I did was to generate flies that were expressing an endogenously tagged POV-M-cherry fusion protein. And I performed live imaging, and I'm showing you here an example movie of a dissected Drosophila testis that's expressing POV-M-cherry to mark the ring canals, and histone GFP to correlate this ring canal formation with the cell cycle. And I'm showing here in the stills, these are stills from this time-lapse movie highlighting a 2-cell cyst with a single ring canal. And following anaphase onset, Pavarotti localizes to the cleavage furrow, and after contractile ring constriction, we see that POV forms this bright mid-body-like intermediate, and this persists for about 20 minutes before POV undergoes an apparent reorganization to form the ring canal. And you can see that clearly here in these insets of ring canal formation and the en-face orientation. And we call this transition the mid-body to ring canal transition. And it differs greatly from the dynamics of cell division or cytokinesis in the somatic stem cell division, where, again, this is a complete division, and following anaphase onset, POV localizes to the cleavage furrow, and after contractile ring constriction, forms these mid-bodies that are very different than the mid-bodies or the mid-body-like structures that we observed in the germ cell divisions, and that they are much smaller in diameter than their germ cell counterparts, and also they do not undergo the same apparent reorganization to form a ring canal. And with the observation of these germ cell mid-bodies, I sought to further characterize these intermediate structures, and I performed additional live imaging of other mid-body and cytokinesis proteins. I'm showing you here stills from a time-lapse movie of testes that were expressing citron kinase, or as in Drosophila, this protein is known as sticky, which is a major mid-body mediating and organizing protein during canonical cell division. And in contrast to what I've already described to you about the dynamics of POV during ring canal formation, sticky remains localized to the cleavage furrow and then the subsequent mid-body ring throughout the entire process of ring canal formation. And it's only when POV undergoes this apparent reorganization do the two proteins then seem to co-localize. And so by imaging sticky as well as anilin, non-muscle myosin, and one of the septins, these data strongly suggest to us that these ring canals are actually the product of a stabilized mid-body ring. And I sought to further investigate the role of citron kinase during incomplete cytokinesis, where I performed RNAi experiments specifically in the germ cells in the testis. And I'm showing you here a panel of control images where ring canals are now labeled with POV-GFP, and I'm also using an antibody to mark the fusome. And in controls, ring canals have a uniform, an average diameter of about 1.5 microns, and the lumens of these ring canals are really obvious. In contrast, depletion of sticky RNAi, or depletion of sticky specifically in the germ cells, results in the formation of these very small ring canals, as you can see quantified here, as well as a high incidence of what we were calling closed ring canals, because there was no obvious lumen, but actually appear more like ectopic mid-body-like foci. And interestingly, these mid-body foci, when they're associated with the fusome is very thin and elongated, and often there are gaps in the fusome, suggesting that cytokinesis was complete. And in fact, further analyses of this phenotype, we find that the level of POV is reduced in these structures, and we show this with both quantification of POV-GFP fluorescence intensity, as well as Western blotting of whole testis lysates. And so these data suggested that citron kinase, or sticky, facilitates this mid-body to ring canal transition, and likely does so indirectly through its role in scaffolding POV protein in these germ cell mid-bodies. And the functional consequence of having an aberrant mid-body is that these mid-bodies impact the communication between these syncytial cells. And so here I was able to perform photoactivation experiments of a photoactivatable GFP protein in intact testes. And here I'm showing you that on one end of the testis, or one end of the germ cell cyst, we'd photoactivate GFP and track its movement to the opposite end of the cyst, and measure how long it takes for the protein to become equilibrated through all cells in the cyst. And in controls, this takes an average of 120 seconds. However, in the sticky RNAi condition, where you can see these very small mid-bodies, the time that it takes to equilibrate photoactivatable GFP protein is significantly delayed, on the order of 500 seconds. And you can see here, directly comparing these 120-second time points, that most of the protein in the sticky RNAi condition is still accumulated around that initial site of photoactivation. And so these results really highlight the significance of the mid-body to ring canal transition in mediating cell-cell communication. And so we published these results earlier this year. And so I want to highlight the major findings. And that is that with extensive live cell imaging, for the first time, we've identified that ring canals form through this extensive reorganization of these germ cell mid-bodies. Importantly, these mid-bodies do not recruit any of the components necessary for abscission, and in fact, undergo this rapid reorganization that's marked by a reduction and a concomitant reorganization of this Pavarotti protein. We show that sticky likely scaffolds POV protein in the germ cell midbody and mediates this midbody to ring canal transition. And again, we highlight that these aberrant midbodies impact the movement of cytoplasmic GFP. And so these results have changed the way that we think about ring canal formation and have provided some really interesting platforms and jumping off points for us to now go in and begin to understand the mechanistic details that mediate this midbody to ring canal transition. And we're using sticky as our genetic entree to begin to understand this molecular mechanism. And so sticky is this multi-domain and very large protein. It has an N-terminal kinase domain, a coil domain, that's shown here split into two halves, an N-terminal and a C-terminal half, a C1 membrane targeting domain, and a CNH or citron-NIC1 homology domain. And working together with Ellery Winkler, a Yale College undergrad, we've now been performing a structure function analysis where we have generated transgenic constructs for overexpression, and I should say transgenic mutant constructs with each of these domains deleted. And in these studies, so far we've only looked in the context of the wild type protein. And so we are looking for things that localize normally to the ring canal, but have a dominant effect in that in terms of sticky function. And so we first generated an endogenously tagged sticky GFP. We show that sticky GFP localizes to the ring canals and to the cytoplasmic puncta. Overexpression of sticky full-length GFP mirrors this localization as the endogenous protein. And also, when we delete the C1 membrane targeting domain, we see that sticky still localizes to the ring canals and to the cytoplasmic puncta. When we delete the C-terminal half of the CC2 domain, we find that this disrupts sticky localization to the ring canal. And instead, sticky then localizes predominantly to the fusome. And our most interesting result was when we deleted the CNH domain, and we find that sticky localizes to ring canals, but we also see an increase in the number of both cytoplasmic as well as ectopic foci that are associated with these fusomes. And we looked at this phenotype a little closer, and we find, again, the formation of these ectopic mid-body-like foci, as well as localization of sticky to these ring canals. So we were excited by this phenotype because it mirrored the phenotype that we saw when we depleted sticky in our RNAi experiments. And this was particularly exciting to us because it's been shown in completely dividing Drosophila S2 cells that the CNH domain plays an important role in binding to the small GTPase row A and maintaining its localization at cleavage furrows. And so we wanted, this suggested that perhaps one way that sticky is mediating or contributing to ring canal formation is by maintaining row A at the ring canal. And so we wanted to investigate the localization of active row A. And so we did this using a row A GTP sensor, which is the row A GTP binding domain of anilin fused to GFP. And here I'm showing you just the tip of the testis where the mitotic divisions are, but I hope that you can appreciate that this anilin row binding domain fused to GFP localizes to the ring canals. And we actually see that this localization persists throughout the entire process of spermatogenesis. And so as row GTP levels typically decline at the end of canonical cytokinesis, this suggests that one possibility is that row A is being atypically regulated during incomplete cytokinesis. And this is a hypothesis that we are now following up on. And so in the future, we're really trying to understand and identify the molecules that facilitate this mid-body to ring canal transition. And this includes investigating two candidates from an RNAi screen that we carried out that I did not have time to talk about. And so we're now generating transgenic tools to begin to look at the localization of these proteins, as well as pursuing this hypothesis of atypical regulation of row A. I'm especially interested in understanding whether or not the mechanism of ring canal formation is conserved across biology, given that ring canals are such an integral part of gametogenesis. And towards this end, I have performed live imaging of mid-bodies or of ring canal formation in the Drosophila ovary and find that this mid-body to ring canal transition is present in the Drosophila ovary and occurs with the same timing or similar timing as in the testis. And also, I find evidence that these germ cell mid-bodies are present in the male germ lines in distantly related species, including the mouse testis, shown here is a mid-body intermediate, and in the cnidarian and non-bilaterian hydrovulgaris testis, where we also find evidence of these mid-body intermediates. And I'm particularly interested in following up on the work in Hydra because it provides a really unique opportunity to begin to ask questions of evolution. And also, ring canals are readily abundant in Hydra. And so we find that ring canals are present in not only cells in the germline, but also in the soma. And 70% or more of cells in an adult Hydra polyp are connected by ring canals. And so we're now performing comparative analyses, just trying to survey the landscape of the ring canal in Hydra using our custom kinesin 6 antibody to label ring canals, as well as commercially available antibodies. And so these have revealed some really interesting insights when we compare them to what's known about the Drosophila ring canal. And that similarly to Drosophila ring canals, both germline and somatic ring canals in Hydra are enriched in phosphotyrosine epitopes and F-actin. But in contrast to what's known about Drosophila, we also find that a subset of somatic ring canals in Hydra also co-label with alpha-tubulin. And so we're using these similarities and differences to begin to inform hypotheses about the evolutionary modifications of ring canal formation. And ultimately, in my future research program, I would really like to understand the functional role that ring canals play in multicellular development and physiology. And I will end by saying thank you to my mentor, Lynn Cooley, and the rest of the Cooley lab, as well as the Yale light and electron microscopy cores, the Drosophila and Hydra communities for critical reagents and technical advice, as well as our neighbor labs at Yale who have provided tissue for my investigations into zebrafish and mouse ring canals, and of course my funding, without which none of this would be possible. And so I will end there, and I would be delighted to answer any questions.
Okay, so we have some questions already. So Bruce Draper asks, great talk, Kari. What effect does sticky KD have on fertility?
The kinase dead mutant, I presume. We have, I have made actually CRISPR mutants, deleting making a kinase dead mutant, and found that actually those animals are homozygous lethal. And so there's clearly an effect developmentally. And so now we're working to generate clones in the germline so that we can begin to understand the effect or the kinase activity and potential role in the formation of ring canals.
Okay, our next question is from Michelle Stars Guyano. Nice work, Kari. For your overexpression of the sticky delta C and H mutants, did you see abnormal germline development? It kind of looked like the fusomes were less branched.
So I have not seen that, and that would be an interesting thing to go back and look for. And so, yeah, I will definitely look for that, yeah.
Okay, so there's another one. So Celeste Berg asks, she says, nice work, Kari. Is sticky also present in somatic ring canals?
Yeah, so that was a bit of a surprise, I guess, to me, because we've shown that the ring canals in the male germline, in the germ cells, it seems to have this very transient localization to the midbody and nascent ring. But when we've looked at the follicle ring canals, we actually see that sticky localizes there. So yeah, we do see it on somatic ring canals. And sorry, I see Bruce's knockdown. So yes, it did affect fertility, but because these are RNAi experiments, it impacted fertility and reduced it, but it didn't completely abrogate it.
Our next question is from Lloyd Court. Have you looked at RHO-A localization and activity in your citron knockdown strain? Because the ring does not open. Would you expect RHO-A to be overactivated and citron acting as a buffering protein?
So I have not looked at RHO-A localization and activity in the context of a sticky RNAi yet. And it's interesting because there are some studies of completely dividing cells in culture where ectopic Rho-A activity actually results in the formation of binucleate cells because they fail to undergo cytokinesis or to finish it. And so that was sort of the hypothesis that we were working on, was that perhaps this ectopic Rho-A activity is functioning in a similar way in the context of the germline.
Okay, our next question is by Howard John Wei. Hi, Kari, great work. I wonder if the mid-body ring canal transition is synchronized across different cells within the same cyst.
Yes, definitely. So all of the cells that are dividing synchronously all have synchronous mid-body ring canal transitions. Yep.
Our next question is by Iris Juanico. Did you know if the ring canals are present only in specific Hydra cell types? Is there any difference in their frequency closer to the head or foot?
Yeah, so ring canals are present in the interstitial stem cells and all of the daughters of that interstitial stem cell. So all of the cells that will go on to make the cnidocytes. And so I haven't seen any difference in the frequency. They really are all over. So anywhere you see a nest of cnidocytes or cnidoblasts that are dividing to generate the nematocysts, you will see that they are connected by ring canals.
Okay, so it seems we'll give one more minute to see if there's any other questions. Leslie Rose says, just a comment, great talk, Kari. So
Thanks, Leslie.
okay, so with that, I would like to thank Yuchuan and Kari for their excellent talks. And this seminar has been recorded and will be available on the SDB website next week. And I also want to invite you to please join us for the next month's seminar on Friday, November 10th, where Nea Alha from the University of Texas Southwestern and Jason Smith from the University of Chicago will present. Thank you everyone for coming.